since it's all Scantron, so pretty much all you need tomorrow. Since our screen is working and our shade is down, we're going to hop back into transcription. And we saw that to get the information in DNA expressed as a final functional protein, an enzyme, some kind of structural protein, a transport protein, whatever your favorite protein might be, is a two-step process. So in the midst of copying a single stretch, a single gene worth of DNA into its sort of intermediate form, which is RNA transcription. And then next week we'll get into, or perhaps even Friday, we'll get into the second part, how the information in RNA then is translated into a protein. And we've already done this DNA replication part already. That's one of the 20 topics on tomorrow's exam. So we've gotten through sort of the general idea. This enzyme called RNA polymerase needs to find the start of a gene, which is called a promoter, which we'll look at in a moment. And then it's going to read one of the strands that we're going to call the template strand and make a copy of the coding strand in RNA. And then it's going to find the other end of the gene called the terminator signal. We'll look at those as well. And there is your piece of RNA that can then be translated into a protein. So the story kind of ends there in bacterial cells, but once we get to this point, we will shift into eukaryotic cells because there are a couple of extra things that happen to those pieces of RNA afterwards. So we will hop into eukaryotic cells eventually. So we saw RNA polymerase has several different subunits, two alphas, a beta prime, an omega, and a sigma. And as you can kind of get the feel from this picture, the sigma can either be attached or detached from the rest. So this part over here on the left is referred to as the core enzyme. When sigma is attached, that was the holo enzyme. So we had talked about all that, I guess, last Friday, I guess. And this details a little bit of what we know that each of the pieces do. And I think that's pretty much where we had gotten last week. Okay. You okay so far? Of course you are. Okay, so first question is, how does the cell, through this enzyme, through this RNA polymerase, know where genes start? Okay, an important idea because, as you might be aware if you've studied anything about the Human Genome Project, it seems that a lot of, at least eukaryotic DNA, doesn't actually code for anything. So there has to be a way to discriminate a section of DNA that codes for something like an enzyme or a protein or whatever, and the parts of DNA that don't, and then also sort of discriminate between one gene and another. You know, the gene for hexokinase versus the gene for, I don't know, actin or something like that. So these sequences called promoters are quite important to get this process started. So we're going to focus on the first type up here in the A section of this slide, where there are sort of two-piece promoter. But there are other types, as you can see here. They're kind of all sort of variations on this basic first type. That's why we're going to focus on this one. But there are several other types that can be found. But if you kind of understand this first type, the others, like I said, are just sort of variations on that. So this first type is sort of the classic bacterial promoter. And of course, again, just to remind you, we're doing the bulk of this process in a bacterial cell. The story in eukaryotic cells, a little bit more complicated. But again, if you have the basic story in a bacterial cell, you'll be able to understand the eukaryotic story if you ever take a course and get that. So what's highlighted in green here is the promoter sequence, and the parts in dark green are sort of the most important pieces of that promoter sequence. So it corresponds to the little purple boxes here in this previous figure. So anything before the start site of the gene, so here's this actual gene start place, so that gets +1. So if you think back to grade school number lines, right? So that's the +1. So anything before that gets a negative number, okay? And that's called upstream of the gene. So anything before the gene is called upstream. Okay, so where the gene starts is +1, and anything before it is called upstream, and that's where promoters are. Promoters are always upstream of their gene. And as people were studying these sequences upstream before the gene actually starts, they saw that pretty much no matter what gene it was, these two sections here that they've highlighted in sort of darker green with the actual letters there were very, 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 very similar from one promoter to the next. So that usually says that that's probably pretty important. Okay, if you see the same sequence over and over again, that's probably indicating that it provides an important function. So of all of this green section, this promoter section, these two particular sequences seem to be the most important. And they are sort of centered at -10s, that means 10 nucleotides before the start. And another section is sort of centered at -35, so 35 nucleotides before the start of the gene. Again, think about number lines. So we have the -10 sequence, the -35 sequence that are shown to you here. So if we look at those two sequences, does anything sort of jump out at you about those two parts of the promoter, those two sequences of those two sections of the promoter? They are, especially this -10 sequence is all AT, so it's 6 of them. TATAAT. And even the -35 of these 6, 4 of them are ATs. Right? 1, 2, 3, 4. So, if you want to make a broad generalization, promoters tend to be AT-rich. We have seen something like that before, haven't we? Nod your heads. You have. You might not remember, although I hope you do because it's on tomorrow's test. Where have we seen an AT-rich sequence before, and what did it do? The origin of replication. What happened there? Right, that was another AT-rich sequence. What happens at the origin of replication? Right, the two strands start to separate. Here we have another AT-rich sequence, again, especially the -10 one, a little bit even more so than the -35. And as we'll see in a moment, the same thing is going to happen at that -10 sequence. This is where the double-stranded DNA is going to start to be pulled apart. And presumably it's AT-rich for the same reason that the origin of replication was AT-rich, because it's easier to pull apart or break apart 2 bonds at a time instead of 3 that you have between G's and C's. So the same sort of idea seems to be in play here. So this promoter then is going to provide a lot of function for this particular gene. Right? Because we kind of already know from the previous pictures, this is where RNA polymerase is going to bind. The sequence that sigma factor, right, that sigma subunit, is looking for. Sigma directs enzyme to promoters. Right? So this is what sigma is looking for. It's looking for this -35 and this -10 sequence specifically. And it's really, really looking for this -10 sequence because it's going to start pulling apart the DNA. And the other major thing it's going to do is tell RNA polymerase which strand it's supposed to be reading. So the placement of the promoter defines what we're going to call the template strand versus the coding strand. Because remember, we're only going to be copying one of the two strands. This is not DNA replication where we pretty much copy everything. We're just copying one of the two strands. So we want to copy the coding strand. That has the actual code in it to make the protein. But remember how weird nucleic acids are, right? If I wanted to make a copy of TATAAT, for example, since that's the coding strand on top, I couldn't read that directly, right? Because if I read a T, what would I put in it? What would I put across from it? An A. I don't want an A, I want a T. So if I want to make an actual copy of the coding strand, which strand do I actually need to read to do that? The template, right? So if I want to make a copy of that, I need to read the complementary sequence, which we're going to call the template strand here. So I read the A, and we put a T. Well, if this is RNA, of course, you put a U, but you know what I'm saying. You read the T, you put the A, you read the A, etc. And so if you want to make a copy of the coding strand, you need to read the template strand. And again, this also gets us back to that basic issue that we always have with nucleic acids. We're going to make the RNA 5' to 3', right? That's always the rule. The same rule with DNA. So if we're going to make the RNA 5' to 3, we're going to read the template DNA in what direction?
3 to 5.
3 to 5. We always go anti-parallel. So all that basic stuff about DNA that we've been dealing with applies here as well. So you'll notice how this is set up. So the promoter is, in this case, to the left of the gene, right? Here's the start site of the gene. The promoter is to the left as we've drawn it. So the bottom strand is the 3 to 5 strand, so that must be the template. And then for the top strand that's running 5 to 3, must be the coding, and that's the one we're going to make a copy of. Because that contains the information to make the protein. Does that kind of make sense? Okay, so that's the importance of this promoter. If you put it somewhere else, you might mess this whole thing up. Right? So it's positioned perfectly so that it knows exactly where the start is. Okay, so if you move this to the left, then it's going to start earlier, which is a bad thing. If you move it to the right, it's going to start to the right, and that's a bad thing. So its position has to be perfect so that the enzyme knows exactly where the start site of the gene is. It knows in this particular example that the bottom strand is the template strand. Okay, so it's going to read that one and make a copy of the top one. So these promoters are really, really, really important. So again, we know that because people have done just those experiments. Okay, so they've taken this, they've moved it, and things have gone awry. They've taken it away, and you don't get transcription anymore. Okay, so they've done all those important kinds of experiments to figure all this stuff out. Okay. So here's the basic idea. This is a little bit more complicated one. This has an extra piece to the promoter, but we don't have to really worry about that. If you kind of ignore that. Here's the -10, here's the -35. Here's that sigma subunit that we've been talking about. And it just so happens that it's pretty much the perfect size to fit that section of the promoter. Isn't that clever? Isn't that convenient? All right, so there's a section that recognizes the -10 region. There's another section that recognizes the -35 region. And then the rest of the subunits are a little bit larger. So these are positioned in such a way that this overhangs the start site of the gene. So it's all kind of about size and location here. This is kind of another way to look at it. So here in yellow is our promoter, and we have our RNA polymerase coming along with sigma attached. So that's an important little factoid as well. For transcription to start correctly, it has to be the holoenzyme. The sigma subunit has to be attached. Why? Because it's the job of sigma to find the promoter. Right? that back from this table. Directs enzyme to the promoter. And so without sigma, transcription doesn't always start correctly. Okay? Can you do transcription without it? Yes. Does it start as accurately? No. Okay, so sigma lends a high degree of accuracy to this process, and it increases the rate at which it occurs, because otherwise, without the sigma, the rest of the RNA polymerase is just kind of wandering around. It doesn't really know where the gene is. It sort of guesses and that kind of stuff. So sigma is really absolutely vital to get this process started correctly, right? Because it has the ability to recognize the promoter, which puts the rest of the enzyme in the perfect location for the start site of the gene, the first nucleotide of the actual gene. So you can see here, so it binds to the promoter and starts opening up the DNA, going from double-stranded to single-stranded, at that -10 sequence, that TAA-TAT sequence, that complete AT sequence. Okay, so again, it's set up that way most likely because that takes less energy than if those 6 nucleotides were all GCs. And so the cell is almost always trying to, sort of save energy if it can. So that's kind of the initiation part. That's how the process gets started. So sigma recognizes the promoter, binds there, opens up the -10 sequence, and by doing all of that, hopefully correctly, it positions the enzyme right at the +1, right at the first nucleotide of the actual gene itself. Does that kind of make sense? And so that's why we need sigma there at the beginning. Once it's done that, though, it's kind of done its job, and it's sort of allowed to leave. Which is a little weird, I guess. It would certainly make it a little bit easier to study if it just stayed attached, right? But we're getting used to weird things in biology, right? So once transcription actually kind of starts, once it starts making that piece of RNA, the sigma subunit detaches, and it's the core enzyme that does the rest of the process. So it kind of looks like we really just need sigma to get the process started, but the other two pieces, elongation and termination, we don't need sigma anymore, so it leaves, physically leaves. So the rest of the process, or at least the middle part, the elongation part, is pretty much just like DNA replication, except you have to remember we're not making DNA, we're making RNA. Okay, so RNA polymerase is wrapping onto RNA nucleotides, not DNA nucleotides. Okay? But the actual process itself is remarkably similar to what we see happening in DNA replication. And this is just kind of showing you some more details about the enzyme itself. And remember we had talked about last Friday, unlike DNA polymerase that needs all these other proteins and all this extra help, RNA polymerase pretty much does the whole job by itself. Right? It can unwind the helix in front of itself. It rewinds it behind it, so we don't need any helicases or topoisomerases. It works on single-stranded DNA, so there's no primase need. Okay, so there's a lot less extra stuff here with transcription. Pretty much RNA polymerase does almost all of it on its own. So that's what they're kind of trying to show us here, where there are sort of pieces of the enzyme that are involved in all of these extra, or all these functions. I thought I had a better picture. Okay. But otherwise, again, it's doing exactly the same thing as DNA replication. It's reading. I'll just draw a little section of a gene here. Here's a little bit of our template strand. RNA polymerase is coming along. And the cycle is pretty much the same as the DNA cycle, except remember we're making a piece of RNA. So it reads the T, it grabs onto N, puts it in place, checks it, it moves to the next location. It reads the A, it grabs a
U.
Making RNA, so it's not a T, it's a U. It makes the phosphodiester bond, it checks it, it moves to the next place. It reads the C, it grabs a G. It makes the phosphodiester bond, it checks it, it takes a step. So it's the same basic cycle that we had for DNA, except it's making a piece of RNA. Okay, so it's going to keep doing this. It reads the G, it puts a C, it reads a G, it puts a C, it reads a C, it puts a G, etc. So the same kind of stuff that was happening for DNA replication is happening here, except we're making a piece of RNA. So again, the clue is RNA. And it's doing it at a pretty good clip. Let's see. Transcription. Not quite as fast as DNA replication, but it's happening about 40 nucleotides per second, give or take. So, not quite as fast as DNA replication. That was like 200 nucleotides per second, so it's a little bit slower, but it's still doing it at a pretty nice clip. So it's doing that cycle 40 times every second. So that's kind of the same. So we had a start. We knew where it started because of the promoter. Along the gene until it gets to, on the other side, some kind of termination signal. Before we do that, actually, everybody okay with the elongation? Does that make sense? Okay. So if we have a start signal, it kind of makes sense to have a stop signal. We need to know where the gene ends, right? Because otherwise it's just going to keep copying, so we need to know where the gene ends. And apparently, there are two major types of termination signals in bacteria. They're both going to end up kind of doing the same thing. They're going to cause transcription to stop happening. So they are going to terminate it. They're going to end the process. There are two ways that this can happen. One of them, I guess this is the one we're starting with, is called row-dependent termination. So in this type of termination, we do need an extra protein, and that protein is called row. So hence row-dependent. It's dependent on row. And row is, as you can see here, a hexameric protein. There are 6 subunits. 1, 2, 3, 4, 5, 6. And the row protein is looking for this particular sequence. That is called, I don't really know why, but it's called a RUT sequence. I'm not exactly sure what that stands for. But in any case, and it has this particular sequence. No, of course, you don't have to memorize that. That would be silly. Okay, but realize row has a specific sequence it's looking for, just like sigma is. So it's the terminator rather than the promoter, and that's the sequence. Again, how do we know that? Because if you delete too much of this sequence, transcription doesn't stop. It just keeps on going. So that kind of suggests that this is the termination sequence. If you take it away, it doesn't stop. Okay, so that's what it's looking for. So we think it kind of works something like this. So the big green blob is the RNA polymerase, and it starts at the promoter and starts transcribing this particular gene. And somewhere early in transcription, the row protein, which is the purple thing here, hops onto the RNA. And apparently just starts scanning along looking for that sequence. As the RNA polymerase is transcribing this gene. And if it finds it, so let's pretend this terminator is a row-dependent terminator, in this example. So if it finds it, as RNA polymerase passes it and transcribes that RUT sequence, that's what row is looking for. It binds there, and somehow, this is the part that I don't think has been worked out completely yet, the interaction between the Rho protein and RNA polymerase, once it's bound to that rut sequence, causes RNA polymerase basically to fall off the gene, is what we think happens. So there are certainly some gaps in our story here. We don't know exactly how all of this works, but for a long time, we didn't know how any of this worked. And all I could say to the class was somehow Rho interacts and it stops transcription. So we know a lot more than we did even like 5 years ago, but we don't know every little tiny detail of this process quite yet. But it seems to work, like I said, something analogous to this general story. So it's kind of an interesting little system. So there's a built-in sequence that's called rut. That's what Rho is looking for. If it finds it, it binds there, and that tells RNA polymerase to fall off the gene, and therefore if it falls off the gene, transcription is sort of by definition over. So that's Rho-dependent termination. Kind of make sense? Okay, so that's one type. The other type is called Rho-independent termination. So this does not require any extra protein, especially Rho. So that's why it's called Rho-independent. This is built into the gene as well. So there's no rut sequence, but there is this particular sequence, which I don't think has a name, at the end of the gene. So here's the Rho-independent terminator. And this is kind of interesting too. So when this terminator sequence is transcribed into RNA, it folds on itself to create this hairpin structure. So we have intramolecular GC binding here, and that folds into this hairpin-like structure. And then you have a stretch of U's in the RNA attached to, or is it here, that stretch of A's in the gene. So here's another picture of the hairpin. They're sometimes a little bit bigger, sometimes they're smaller, but there's always some kind of hairpin involved. Followed by a stretch of U's in the RNA attached to a stretch of A's on the DNA. So what we think happens is something like this. So they haven't drawn RNA polymerase into this. So RNA polymerase is basically here. It's just transcribed through the terminator sequence, and here is that stretch of U's. Okay. The terminator sequence folds into that hairpin, and here's RNA polymerase here. And somehow, we don't completely understand this either, somehow that hairpin structure seems to interact with RNA polymerase, kind of like Rho did, and it sort of taps it on the shoulder and says, okay, we're done now. And it causes RNA polymerase to fall off. And since the only thing that's keeping the RNA attached to the DNA is this little stretch of U's attached to the A's in the DNA, and we already know there's only 2 bonds between each one, that's going to be more fragile. So once RNA polymerase falls off, this comes off, and transcription is done. So this one's even kind of nicer in that there's no extra protein involved. It's only sequence dependent, and this particular sequence, like I said, forms into this hairpin. Somehow that hairpin interacts with RNA polymerase and says, okay, we're done now. Okay, so certainly there are details that we don't have yet. But I sort of like that aspect that we don't understand everything yet. Because I think we all sit in these classes and say, we look at the thickness of our textbooks and we think we know everything there is to know. We do not. Okay, so while we know the sort of general stories of how transcriptional termination stops, we don't know every single last little detail. But this is the sort of the general story there. So this actually might be a little better picture. Okay, so here's the enzyme, here's the hairpin, there's the little connection between the RNA and the DNA. So again, somehow that hairpin is interacting with the RNA polymerase and saying, okay, we're done, you can release me, and you can fall off the DNA. So it's an inherent part of the gene itself, so you don't need any extra protein. So I've never been able to find a good paper that tells like how many Rho-dependent versus how many Rho-independent genes there are. I've never come across a paper that kind of gives me numbers for that. So I don't know what the breakdown is They're the two different kinds of terminators, but they are both present in most bacterial cells. They both kind of make sense? You see the subtle differences between the two? Take silence for yes this morning. Okay, so that kind of in a nutshell is transcription. Let's show you the animation. Now this animation is a little overly simplistic based on what we just talked about, but it will show you at least the general features.
The first step in protein synthesis is transcription of RNA from DNA. The transcription of RNA from DNA begins with initiation. An enzyme called RNA polymerase attaches to a region of the DNA called the promoter, which lies next to the beginning of a gene. RNA polymerase pries the DNA strands apart, and RNA nucleotides line up along only one of the DNA strands, the template strand. RNA polymerase links the nucleotides together. The second stage of transcription is elongation. RNA polymerase moves along the DNA, adding nucleotides to the end of the growing RNA molecule. The RNA strand peels away, allowing the two separated DNA strands to come back together. Here's a close-up view of elongation. RNA nucleotides pair up with their complementary DNA bases on the template strand. Note that U in RNA pairs with A in DNA.
If we look closely here, right, we're reading the template strand, and what are we making a copy of? Coding strand, right? G with G, C with C, A. Again, we swap T's for U's, but they kind of mean the same thing, right? So by reading the template, we make a copy of the coding strand of the DNA. That's kind of the whole point.
RNA polymerase moves along the DNA strand, linking nucleotides to the end of the RNA strand. The last stage of transcription is termination. Once RNA polymerase has gone past a sequence called the terminator, the enzyme releases the completed RNA and detaches from the DNA.
So that's the very simplistic part. So I guess since they didn't show us another protein, we can assume this one is a row-independent terminator, but they don't show us the hairpin or anything like that, but you get the general idea from this. So, like I said, a little bit overly simplistic, but it will do a pinch. So that's sort of the basic story of transcription in bacterial cells. We do need to now hop into a eukaryotic cell because there are things that are going to happen to that piece of RNA in our cells, in eukaryotic cells, that do not happen in bacterial cells. So we do need to kind of make the shift and look at the product of a eukaryotic gene and then look at what are called post-transcriptional modifications. So these are for eukaryotic cells only. So that's why we have to make this sort of transition. So in eukaryotic cells, there are a series of post-transcriptional modifications. First of which is starting to be shown here is going to be the removal of sequences that are called introns. So first we need to sort of talk about what they are and why we think they exist and how we get rid of them. So if you look at a typical bacterial gene, like at the top here, there are no, or at least there are very rarely any of these intronic sequences. So I guess we'll start with the definition. So what's an intron? It is surprisingly a non-coding section of the gene. Whereas what we're going to call an exon is an actual coding section of the gene. And they sort of alternate, as you can kind of see in the bottom part of this figure. So here's a typical eukaryotic gene. The exons are the dark orange, and the introns are sort of the lighter tannish color. So you can see they sort of alternate. So exon, intron, exon, intron, exon, intron, exon, intron, exon, intron, exon. So there's always an exon on either side, okay, with introns in between. So there's always one fewer intron than there are exons in your gene. And RNA polymerase can't tell the difference between the two. Okay? All RNA polymerase can do is just copy everything. Okay? So there's the promoter, there's the terminator, everything in between RNA polymerase copies. Okay? So RNA polymerase cannot tell the difference between an intron and an exon. And most eukaryotic genes have at least one intron. Some have many more. Okay? So the beta globin gene that makes your part of your hemoglobin looks like it has 3 exons and 2 introns. The human factor VIII gene, which is involved in blood clotting, looks like it has 26 exons and 25 introns. And there's the table I wanted. Whereas actin, actin and myosin that lets your muscles move, only has 1 intron. This gigantic gene called Titin has 233 introns, so runs the whole gamut. Okay, so different genes of different sizes will have different numbers of introns, but you see in each case it's always 1 fewer intron than you have exons. And what we're going to see is that the introns are going to have to be removed if you want to make the actual protein. Okay, so since the introns don't actually code for protein, you kind of need to get rid of them. And there's going to be a whole set of machinery that we're going to look at in just a moment that will do this, what's called splicing of the introns, splicing out of the introns. That first. I'm sorry, let me just jump ahead just quickly. Okay, yeah, we'll do that later. Okay. So we'll look at the mechanism first, and then we'll kind of backtrack a little bit and think a little bit about why these things even might exist, because it seems a little silly. It might seem a little silly at first glance. Okay, so let's look at how they're removed first. So, like I said, the RNA polymerase doesn't know the difference. It just reads, starts at the promoter, ends at the terminator, and copies everything. So the initial product, the initial RNA product, is sometimes referred to as a pre-mRNA. Another term you might come across is the primary transcript. Is another term that I actually like a lot. It's called primary transcript or pre-mRNA. Whatever you want to call it, it has both exons and introns in it. Because that's all RNA polymerase can do. It just copies, it just makes RNA. But what we need to happen is sort of twofold. We need to splice out the introns and then glue together the exons. All right, so eventually this is going to shorten if we pull out the 2 exons and glue the other introns together. No, backwards. If we get rid of the introns and glue together the 3 exons, then our mature messenger RNA is going to be shorter than that primary transcript, right? Because we're going to splice out the introns. And that's initially how these were discovered. People noticed that the RNA, while it was still in the nucleus, was a lot longer than the RNAs that came out into the cytoplasm and got translated. And then the rest of the story was sort of pieced together over several years. does physically get shorter, okay, because the mature messenger RNA that's eventually going to get translated into a protein lacks introns. It lacks these sequences that actually don't code for protein. Okay, so this is just kind of another color-coded picture for this. So again, we want to get rid of the blue introns. So here's our primary transcript. If we splice out the blue intron and we splice together the yellow and the green exon, we splice out the second intron and splice together the green to the red exon. So again, our primary transcript is longer than our mature messenger RNA. So it turns out there's a whole system that has to be put in place to make this happen. And the first part of the system is set up to ensure that the cuts that are made in exactly the right place. Right? Remember the whole setup of the genetic code, right, is based on triplets, right? There are reading frames. Remember that from last week when we looked at the genetic code. So, if we don't cut these introns exactly correctly, then we're either going to maybe cut out a piece of the exon, that would be bad, or leave in a piece of the intron, also would be bad because it would mess up the reading frame of the whole gene. So these introns have to be spliced out absolutely perfectly every single time. So people started looking at the boundaries between an exon and an intron on one side, and then between the intron and the next exon on the other side. And guess what they found? They found specific sequences. Okay? So we've seen origins of replication as special sequences. We've now seen promoters and terminators. Now, the sequences at an exon-intron boundary are very important to sequences and are very, very consistent in the sequence. Well, again, once you see that, then you say, okay, now we know what the intron-exon boundary is because it's always this, and here's the intron-exon boundary because it's always that sequence. So this is showing you what those sequences are. Again, I don't need you to memorize them for this class at least. But realize that there are specific sequences on either side of the boundary that the machinery is going to be looking for. And as you can see here, from exon into the exon always ends with AG, and then the first part of the intron is GU, R means purine, so GU pyrimidine. And on the other side is always CAG, and the intron leading into a G in the next exon. And what the machinery is going to do is cut between the G's in both cases, between the two G's. And then that G from that first exon will get glued to that G in the second exon that we have here. The machines that do this are called spliceosomes. Oh, actually, this just shows the process. It's kind of a two-step process if you like reaction mechanisms. So first thing that happens is that it breaks, first part here, it breaks that phosphodiester bond right there between the two G's there. And then connects the first part of the intron sort of to an internal adenine there and creates what's sometimes referred to as a lariat. Think about cowboys. So it looks like a lariat. And then, in the second part, it takes the first exon, glues it to the second exon, and by doing so, cuts out the other half of the intron. So here's our intron, and there's our two glued together exons. Again, we're not going to get all that deep into the actual organic chemistry of this, but it is just organic chemistry. But what happens is that we glue together the two exons, we remove the intron. And presumably, this intronic piece of RNA is broken back down into nucleotides and they're reused. That's most likely true. There we go. Okay, so what's doing all of this? What's doing all this in the aggregate is a large multi-protein complex called a spliceosome. And a spliceosome is made up in turn of smaller things called SNRPs. I know you're sitting there saying, who the heck makes up these names? Not me. Okay. So each of these little circles here is a SNRP. SNRNP. I always put the U in just so you can say it there. Okay, so that's SNRP, that's SNRP. And SNRPs are interesting because they consist of both protein and RNA. So I think this is our second example of this. What was our first? We've seen something else was made up of protein plus RNA. No, nucleosomes are just protein around which you wrap DNA. But we've seen something. Not yet. So we haven't actually looked at those yet. We will next week. Right. Telomerase was an enzyme made up of RNA and protein. So here's our second example. Ribosomes will be our third when we get to those. Okay. So this is no longer an isolated idea. There are lots of things in the cell that are made up of part protein, part RNA. So these SNRPs are another example of that. That's actually fast. I have 2 more minutes. Okay. The spliceosome is an aggregation of individual SNRPs. Okay, so by the time we get to this section here, this is the spliceosome. Okay, that's consisted of all of these different SNRPs. And the different SNRPs presumably are doing different roles in this process. Okay, so maybe some of them recognize that first junction, another one cuts it, a third one recognizes or creates the lariat, a fourth one recognizes the other junction, the fifth one, makes the second cut, those kinds of ideas. That's probably why we need so many of them. But it is the job then of all of these SNRPs coming together as the spliceosome is to recognize the exon-intron boundary, snip out the intron perfectly, glue together the 2 exons perfectly every time. Because any imperfection is going to mess things up pretty royally. If you leave in an extra nucleotide or you cut out an extra nucleotide that you're not supposed to, is going to be very, very bad. That's going to create what we call a frame shift mutation. Okay? Don't want those. It needs to be perfect every single time. Okay, so we will stop here for today. Okay, we have our test tomorrow, so please be on time to lab if you need a pencil.